Welcome to VMJ Shorts, where we discuss the latest articles in the Vascular Medicine Journal with authors and experts in the field. I'm Polka Chaudhry, and today I'm excited to have Doctors Anu Aggarwal and Scott Cameron discuss their latest manuscript on dysregulated platelet function in patients with post-acute sequelae of COVID-19. Dr. Anu Aggarwal is a post-doctoral fellow at the Cleveland Clinic Lerner Research Institute, and Dr. Scott Cameron is a platelet biologist and the section head of vascular medicine at Cleveland Clinic. Welcome to both of you, and thank you for joining us.
Thank you for having us.
Thanks so much.
Let's start with long COVID, or the post-acute sequelae of SARS-CoV-2. This entity seemingly has affected many patients after the initial infection with COVID-19, and yet it is poorly understood. Can you explain what this clinical entity is?
Post-acute sequelae of COVID-19 is recognized as multisystemic condition that is characterized by persistent symptoms beyond 4 to 12 weeks after the initial COVID-19 infection. So these patients are labeled as long COVID by the Center for Disease Control and National Institute of Health. These long COVID symptoms typically include fatigue, dyspnea, chest pain, headache, cough, and difficulty breathing.
Tell us, why did you choose to study platelets in this patient population, and what were your major findings?
Platelets play an important function in preventing excessive bleeding, that's hemostasis, alongside an important role in the host immune response to invading viral and microbial infections. So, one study from Michelle Group revealed the protective effect of this oral factor 10A inhibitor on discharged patients by protecting them from deep vein thrombosis. So, this led us to study both platelet function and thrombotic potential in these patients. The interesting findings of our paper is when we spike the platelet deplete plasma from these long COVID patients to the washed healthy platelets, and there was surprisingly profound activation of platelets after the stimulation with platelet receptor agonists. And also, in all these patients, through platelet aggregometry studies, we found they have higher platelet aggregation through thromboxane receptor. So these were quite interesting studies that we found even after 16 months of COVID-19 infection, they still had a dysregulated platelet function.
The biggest surprise, I guess, that both of us had is that contrary to patients with COVID-19, which clearly has a thrombotic milieu, there have been maybe some case reports of patients with long COVID that may have something similar, but what we found was either no difference or actual underactivity of platelet function by light transmission aggregometry. And it's pretty clear that the platelets that are budding from the megakaryocyte in the bone marrow in patients in long COVID are probably not the same kind of platelet. They've somehow been reprogrammed, perhaps to try and adapt to the increased thrombosis that one might see in COVID.
Dr. Cameron, what do you think is the major significance of these findings, and what are the big picture implications of these results?
The first thing we'd have to obviously say on the record is, it was a small study of about 30 patients with long COVID and fairly matched controls. We certainly wouldn't want to generalize it to the population at large. But one of the first issues I thought about is if somebody had recovered from COVID and they do have symptoms that are consistent with post-acute sequelae of COVID, should those patients be blood donors? Should they be plasma donors? Their plasma may be slightly different. The second thing, and I think one thing that Dr. Aggarwal showed pretty clearly in a small sample by light transmission aggregometry, signaling through the thromboxane receptor seemed to be augmented. And now that's a pathway that you would suppress with aspirin. Thromboxane production by the platelets decreased with aspirin.
Lastly, I noted that the overwhelming majority of patients enrolled in your study are women. And as a scientific community, we have famously done a very poor job of enrolling women in studies in the past. Tell me about your approach and why enrolling women was particularly important in this study.
Interestingly, as you asked, so mostly whenever we do any study in our lab, We try to include both men and women in our patient cohort, and as well as in the controls, because we also work with mouse samples. We have noticed that there is a difference in platelet reactivity in this male and female mice as well as human samples. But specifically for long COVID, in the literature, according to CDC survey, the long COVID affects nearly twice as many women as men, with approximately 6.6% of women getting affected with long COVID, and only 4% of men were getting affected after COVID infection. So I would really like to thank our coordinators because they did a great job of collecting all the women's samples.
Dr. Aggarwal and Dr. Cameron, thank you so much for your time and sharing your data with us. And thank you, viewers, for your time. You can check out this article and all the other latest articles by clicking the link pinned to our X profile page.